I'm Austin.
I'm Keith.
I'm Remy. And this is cloning. It's just a thing made from another thing that has the exact same genes as the original. Essentially, it's a copy. And cloning has been going on in nature for thousands of years. Identical twins, plants that self-pollinate, and when you cut a starfish in half and it grows a new body, that's all cloning. But the ability to clone something on a cellular level is a very new concept that hasn't been along for a very long time.
Hi, I'm Peter. We'll be talking about early progress in cloning. The first cloned animals were created by a scientist named Hans Dries in the 1800s. Dries' original goal was not to create identical animals, but to prove that genetic material is lost during cell division. Dries' experiments involved sea urchins, which he picked because they have a large embryo cells and grow independently of their mothers. Dries took two-celled embryo of a sea urchin and shook it in a beaker full of seawater until the two cells separated. Each grew independently and formed a separate whole sea urchin. In 1902, another scientist, embryologist Hans Spemann, used a hair from his infant son as a knife to separate a two-celled embryo of a salamander, which also grew externally. He later separated a single cell from a 16-celled embryo. In these experiments, both large and small embryo developed into identical adult salamanders. Spemann went on to propose what he called a fantastical experiment to remove the genetic material from an adult's male and use to grow another adult. In this way, he theorized he was able to prove that no genetic material was lost as cells grew and divided.
In 1986, two teams, one led by Steen Willadis, some British guy, claimed that he had cloned a sheep embryo. But at the same time, on the other side of the earth, in America, not the other side of the earth, but in America, this guy named Neil First had claimed that him and his team had cloned a cow embryo. And, many advances had been made in this time on cloning, and they just a huge step. They went from, point C to point B. But because they didn't think that you could ever take an adult cell and clone it and create a whole new animal, they gave up, and the whole idea of cloning fell by the wayside.
Ian Wilmot at the Rosalind Institute in Scotland was assigned to a project in 1986. His goal was to create a sheep that produced a certain chemical in its milk. He began paperwork in 1987 and began research in 1990. One of Wilmot's colleagues, who had experience with cloning from early embryo cells, suggested that the reason so many cloning attempts failed was that the cells were in incompatible stages of life. On July 5th, 1996, a lamb was born, cloned from a frozen mammary cell from another adult sheep. Dolly took 277 tries to create, and other labs were unable to reproduce the results.
Reliable cloning can be used to make farming more productive by replicating the best animals. It can make medical testing more accurate by providing test subjects that all react the same way to the same drug. It can allow mass production of genetically altered animals, plants, and bacteria. It may settle once and for all what part of personality is dependent on genetics and what part on environment. In short, it can be beneficial to almost every area of biological science. It can make more hamburgers.
So, how does cloning work? The circuit to cloning lies in removing an egg cell's nucleus and replacing it with the nucleus of a donor cell. The egg cell, which has a clean state and is prompt to use information in the DNA to create a new organism. Now, the DNA of the donor organism, however, this transfer is a very difficult process, more difficult in some cases than others.
So, you might know a little bit about how cloning works. Here's the full story. See, the first cloned animals using the modern method of nuclear transfer were frog embryos, cloned in 1951. The reason frog embryos were cloned long before any other mammal is twofold. First, the cells of an 8-cell frog embryo are huge compared to a mammal. You can almost see them with the naked eye. And so obviously they're going to be a lot easier to work with than the tiny little mammal cells that we're used to. And secondly, frog embryos can grow in lab conditions much, much better and much easier than a mammal because, it's a frog and it's supposed to grow up in a pond without a mother. But as far as a mammal, it has to have the nurture and the care of a mother, which it's kind of weird for scientists to do that. and so it doesn't really work out. Anyway, so although happy about cloned frogs because scientists are like that's pretty cool. Though happy about that, scientists they wanted to see cloned mammals because mammals, humans are mammals. If we can clone humans, that's going to be legit. So they did anything they knew about the amphibians and the frogs and applied it to the mammals. And so unfortunately, because of the difficulties associated with mammal cloning, this is a difficult task. Techniques to make this task possible were developed at the labs of Neil First and Steen Willadesen. It's weird. Anyway, those, the two guys mentioned earlier, they worked independently to clone embryos. Once the nucleus is transferred from the donor cell into the egg cell, the nutrient in the solution is put in until it divides into an 8-celled embryo. When the cell passes this point, the individual cells begin to differentiate. One cell may eventually form the head, another the torso, some the arms, some the legs. At this point, the scientists have to quickly move and put the embryo into an artificially sustained oviduct of a sheep or other animal. This oviduct functions as an incubator to nurture the embryo through this critical period. Once the embryo is large enough to be able to withstand procedure, it is placed in a living surrogate mother, where it will be carried to term, nourished by an umbilical cord. So basically, they take the embryo that they had in the oviduct, which is like a fetus kind of thing. It's where it's like a incubator. Once they get that, then they put it in a living surrogate mother. Shut up. They put it in the surrogate mother, and then that way it can grow just like it's in a real baby. Okay? With all these steps, it's no wonder that the success rate for cloning is low. Embryos die each step because there's tons of steps. You got to move from here to here to here. The clones, the embryos, they die at each step, and it's just really, really hard. So that's why cloning is a very new and hard concept to do. And now for the cons. Oh, hey there. One con of cloning would probably be that you have to go through all these tasks and all these obstacles to get the clone in the first place. You're moving it all around. It's really hard to make a clone. So basically, the input, the cost that it takes to make the clone is more than the receiving end. So you put in more, you get less. What's the point,
Another con of cloning is that the animal dies quickly and doesn't get very far in life.
Another con of cloning is that in the future we might have an overpopulation problem, and then we'd have to start killing off clones.
We're gonna do some stuff. You know what?
You can stop for.
In class, will you give us like some minutes?
And now for the pros. You filming? Okay. One of the pros of cloning would be that you create a very vast, powerful, and loyal army, much like in Star Wars, to help fight your wars. Another one being that you could create body parts, limbs, hearts, kidneys, things of that nature, to help out people who have been injured in wars or maybe had a heart failure or something of that nature. Another pro being that you could create vast quantities of burgers, because burgers are good. And the last pro being that cloning is just really, really cool. This one's for Uncle John. I love you. Let's go. Come on.
To all my friends, present, past, and beyond, especially those who weren't with us too long, life is the most precious thing we could lose. While you were here, the fun was never ending. Love a minute was only beginning. Arson's Uncle John, this one's for you! Oi, 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 oi. Remember whose side it is that you're on. You've got friends with you till the end. If you're ever in a tough situation, we'll be there with no hesitation. Whoa.